Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CSIR Net Life Science JRF question paper. This is part C discussion, and I will be discussing cell signaling and immunology in this, which was asked in shift 1 or the morning shift question paper, and this was conducted in the year March 2025. So coming to the first question. The statements below are made about some animal pathogens that may evade innate and inflammatory response by following mechanisms. Okay. So some animal pathogens are there. They can evade the innate and inflammatory response by modifying some of their proteins. So what do they do? Or through some mechanisms. Okay. So you have to tell whether their modification will be the correct way. Whatever they have modified, whether it is correct or not. Okay. So the first one, the flagellin of proteobacteria has a mutation that prevents it from being recognized by toll-like receptors. Okay. Now flagellin, it is recognized by the TLR5 or toll-like receptor 5 that is involved. Now if the flagellin is mutated as in proteobacteria, TLR5 will not recognize it, and it will prevent it from evading and it will help the proteobacteria to evade the innate and inflammatory response. So A is correct. A is correct means 1 gone, 3 gone. 2 and 4 we have to see whether B or C is correct. Okay, the lipopolysaccharides of Helicobacter have mutation that prevent it from being recognized by the TLR. Okay, lipopolysaccharides are recognized by TLR4. So if lipopolysaccharides are mutated as in case of Helicobacter, naturally it will not be recognized by TLR4. And it can evade the innate and inflammatory response. So that makes B also correct, and your answer is going to be option 4, A and B. Okay, we will see others also I will tell. M. tuberculosis escape from the phagosome to the cytosol. No. Mycobacterium tuberculosis, it has got the ability to block phagosome maturation and prevent the phagosome from fusion with the lysosome. So that is how it avoids the lysosomal degrading enzymes, not by escaping from phagosome to the cytosol. So that is wrong. So that makes 2 as the wrong answer, and your right answer is for A and B. Okay, the other one, influenza virus produce YOP protein. No, YOP protein is not produced by influenza virus. It is PB1F2 and NS1 that inhibits inflammasome activity. Then Salmonella typhi encodes a protein that binds type 1 interferons and prevent them from binding to the interferon receptor. It's also not correct. So your answer is A and B. Okay, the next one. The insulin receptor is a receptor tyrosine kinase. Okay, that means it can phosphorylate and engages the PI3 kinase pathway. Okay, so PI3 kinase AKT pathway, that is it, to regulate FOXO transcription factor. So FOXO is a transcription factor and it is involved in the transcription of genes. A student uses QRT-PCR to determine the expression of a direct FOXO target gene, gene X. So here the student is using quantitative real-time PCR to determine the expression of gene X, and gene X will be bound by the transcription factor FOXO. Okay. So whether FOXO is binding to gene X, transcription take place. If FOXO is not binding to gene X, transcription will not take place. So that the student can measure by QRT-PCR. Whether the gene is transcribed, you will get a curve. If it is not transcribed, you will not get a curve, like that. Just example I told. in a mammalian cell line under different conditions and makes the following observation. So which are the correct statements? So what is this PI3K AKT pathway? So this is your insulin receptor. This is your insulin receptor. When it is bound by insulin, the PI3 kinase is activated and it will phosphorylate PIP2 and convert it to PIP3. PIP3 activate PDK1, PDK1 phosphorylate AKT, AKT is activated and it will phosphorylate FOXO, FOXO will be this phosphorylation of FOXO will help in the binding of the 14-3-3 protein, and this binding of this 14-3-3 protein will inhibit the FOXO from entering into the nucleus and result in the transcription of the genes. FOXO is a transcription factor, so it has to enter the nucleus and bind to the DNA to result in the transcription of genes. Now FOXO is prevented from entering nucleus means it will not be able to bind to that gene. There won't be transcription of that gene. So that is what is taking place in this PIP3 AKT pathway. Now what is the role of P10 and FOXO? Now P10, it is a tumor suppressor gene and it removes the phosphate from molecules. So when P10 is added, PIP3 phosphate is removed and it is converted back to PIP2. So by acting that way, it inhibits the PI3K AKT pathway. So inhibition of that pathway means AKT is not phosphorylated. AKT not phosphorylated means FOXO not phosphorylated. FOXO happily enters into the nucleus. It is a transcription factor. It will bind to the DNA and it will result in the transcription of genes. This is what is happening here. Now see the options. Treating cells with P10 inhibitor increases gene X expression. Now you are treating the cell with a P10 inhibitor. Now P10, it inhibits the PI3K AKT pathway. I told you. Now P10 is inhibited means PI3K AKT pathway is active. So AKT is phosphorylated. AKT phosphorylated means FOXO is phosphorylated. FOXO is phosphorylated means it cannot enter into the nucleus and act as a transcription factor. So the translation and transcription of gene X is reduced. That is, gene X expression decreases. So it is not increases as per the question. It has to be decreases. So A is not correct. So that makes option 1 wrong and option 4 also wrong. Okay, option 2 also is wrong. Oh, fine. Then your answer is B and C only. Okay, we will see. A cell line with AKT S308A mutation. So S308 is the amino acid which get phosphorylated in AKT. Okay. Now that amino acid itself is mutated means has increased gene X expression, of course. Because the amino acid is mutated, it cannot phosphorylate. PDK1 cannot phosphorylate AKT. AKT is not phosphorylated means FOXO is happy. It is not phosphorylated. It is entering into the nucleus, transcription of genes. So increased gene X expression. So B is correct. Then change in gene X expression due to the ligand binding defective insulin receptor is partly reversed by a P10 inhibitor. Okay, that's also correct. What they are telling is change in gene X expression. How? Due to ligand binding defective insulin receptor. Okay. So what is happening when this receptor itself is defective such that it cannot bind insulin? Okay. PI3K AKT pathway is inhibited. FOXO will not be phosphorylated. Gene X expression take place. Now what they are telling is this change in gene expression. Okay. Due to this defective insulin receptor is partly reversed by a P10 inhibitor. So, defective insulin receptor, gene X expression take place. Now, inhibiting P10, you are inhibiting P10, PI3K kinase AKT pathway is active, AKT is phosphorylated, FOXO gets phosphorylated, it will not be able to enter into the nucleus and act as transcription factor. So, gene X expression reduces. So, gene X expression due to ligand binding defective insulin is reversed by P10 inhibitor. Ligand binding defective insulin receptor, gene expression is active more. P10 inhibitor expression is less. So, from more to less, activity reversed by P10 inhibitor. So, C is correct. The next one, phosphorylation of FOXO by PDK1. Okay, PDK1 doesn't phosphorylate FOXO. It is AKT that is phosphorylating FOXO. So that makes D itself wrong. So the answer here is B and C only. Okay, the following statements are made regarding the role of cadherins in cell junctions. Okay, so cell-cell, what is that? Cell-to-cell adhesion is by cadherins. Okay. So cadherins are calcium dependent adhesion molecules. Yes, they require calcium ions to function. That mediates cell-to-cell adhesion. Okay, cell-to-cell adhesion. Two cells nearby, they both can be cell-cell adhesion can take place because of cadherins. And how do cadherins cause this cell-cell adhesion? By forming homophilic interaction. What is this homophilic interaction? Cadherin bind to other cadherins of the same type on nearby cells, forming homophilic interaction. So, one cadherin of this cell will bind to the cadherin of the same type on an adjacent cell, forming homophilic, same type interaction. So, A is correct. So, that means 1 wrong. Okay, 2, 3, 4, A is there. Okay, we will see. Then cadherins function in tight junctions. No, they are found in adherence junctions, not in tight junctions. So, tight junctions, it is proteins like claudins and occultins. So, that makes B wrong and option 2 also not correct. N-cadherins are found in epithelial cells. No, N-cadherins are found in nerve cells, not in epithelial cells. Epithelial cells have E-cadherins. So that makes C also wrong. Okay, they have not given that in options. Okay, C also wrong. Then cadherins are involved in the formation of focal adhesion. So focal adhesions are the site of attachment of cell to the extracellular matrix. That is focal adhesions, and it is the main component of focal adhesion is your integrin proteins, not cadherins. So D also wrong, that makes option 3 wrong. Then cadherins interact with actin cytoskeleton through associated proteins like catenins, that is correct. Okay, 4 is correct. A and E are correct. Then next one, a researcher placed plant cells in hypertonic solution, so they get plasmolysed to cause cells to shrink. Yes, the protoplast detaches from the cell wall. The following statements are made regarding the plasmolysed cells. Okay, which are the correct statements? Microtubules are lost in the plasmolysed cells. Microtubules are never lost in plasmolysed cells. Okay, they can undergo some reorganization or something like that, but not lost. So that makes option A as wrong. I mean statement A as wrong. And options 1 also wrong, 3 also wrong. Oh, B is correct. Okay, fine. Protoplasts are retracted in the plasmolized cells. Of course, you are learning from your high school. So B is correct. Then Hectian strands are lost during plasmolysis. So what are these Hectian strands? So this is your cell wall. This is your protoplast, which is retracting from the cell wall when placed in hypertonic solution. This is nucleus. This is vacuole. So this is a strand of cytoplasm that is attached between the contracted protoplast and the cell wall. That is called as Hectian strands. Okay. So they are lost during plasmolysis. They are rather visible during in a plasmolyzed cell. So C is not correct. So that makes 2 also not correct. Option 4 will be your answer. Patches of plasma membrane remain affixed to the cell wall. That is what I told you just now. So D also correct. So that makes option 4, B and D correct. Then next one is regarding apoptosis in C. elegans. Okay, following statements are made regarding apoptosis in the nematode C. elegans. You have to find the one which represent all correct statements. So when in C. elegans, when it receives apoptotic signals, the EGL1 activates and it will inhibit the CED9, which is an anti-apoptotic protein, and in the absence of apoptotic signals, CED9 is found bound to CED4. Now when it gets inhibited by EGL1, CED9 releases CED4. CED4 then activates CED3 and result in apoptosis. This is what is taking place. Now see the options. The human ortholog of C. elegans, CED9. So which is the human ortholog of the C. elegans CED9? It is BCL2. CED9 and BCL2, they are inhibit apoptosis. That is, they are anti-apoptotic proteins. It is overexpressed in B cell lymphoma. Natural, it is a cancer, so it will be overexpressed. So A is correct. So that makes option 4 wrong. Okay, A, CED9 gain of function and CED3 loss of function double mutant will have more than 947 non-gonadal cells. That is correct because CED9 anti-apoptotic protein, CED3 protein essential for apoptosis. Now double mutant means it has CED9 gain of function and CED3 loss of function mutation. So CED9 gain of function mutation means cell is protected from death. There is no programmed cell death at all. CED9 is gain of function, anti-apoptotic protein. Cell is not dying. Okay, the cell is even more happy. It has got CED3 loss of function. CED3 is not going to act. Loss of function, so no apoptosis at all. Okay. Now so there is no apoptosis in double mutant. Now what is happening? Now normally 131 cells undergo apoptosis during C. elegans development. And after this apoptosis, there will be 947 cells left in the adult. Now because of this mutation, it will result in no apoptosis is taking place in the double mutant. So naturally there will be more than 947 non-gonadal cells in this double mutant. Okay, so B is correct. Okay, B is there in 1, 2 and 3. Okay, now C for C and D. If purified EGL1 is added to the CED9 CED4 complex in vitro, the autocleavage of CED3 does not occur. See, EGL1 is added, CED9 CED4 association is disrupted, CED4 is free to phosphorylate or what is that? Autocleavage of CED3 will occur. So it should be autocleavage of CED3 occurs, does not occur. Okay, so C is incorrect statement. So that means 1 gone. Now we have to see whether D is also correct. CED8 is a multi-spanning plasma membrane protein that is required for externalization of phosphatidylserine. Okay, you know that in during apoptosis, if the cell is going to go apoptosis, it will send its phosphatidylserine on the external side of the plasma membrane. It is a signal telling eat me. Okay, so it is calling the phagocytic cells to come and phagocytose the cell, resulting in apoptosis. So CED8 is the one which is acting to externalize this phosphatidylserine in C elegans. So D is correct. So more correct answer is 3, that is A, B, D only. 2 is not correct. They have not mentioned D there. So 3 is correct answer. Then next one. Lethally irradiated mice were split into 4 groups and experiments were conducted as described below. So what is this lethally irradiated mice? What will happen to that? So this lethally irradiated mice means they have received sufficient radiation in such a way that their hematopoietic stem cell failure has happened. That means no immune cells are produced in such mice and they will die very fast. So you know hematopoietic stem cells, they give rise to different blood cells, that is through the myeloid and the lymphoid lineages. Myeloid cells lineage will give rise to the monocyte, macrophage, neutrophil, eosinophil, etc. And the lymphoid cells, it will give rise to T cell, B cell, NK cell. Now hematopoietic stem cells, they reside in the bone marrow. So bone marrow hematopoietic stem cells can differentiate into B cells, T cells. Remember one thing, T cells originate in the bone marrow but mature in the thymus. So for T cell to form, bone marrow has to be there. Okay. So now what they have done? They have done different experiments, that is they have given cells to various mice which are lethally irradiated. Okay, they have given thymus derived cells, they have given bone marrow cells, they have not given any cells also. So what is the outcome of this experiment? You have to tell. So 4 possible outcomes listed below were suggested. Group 1 mice are unlikely to survive. Now what is group 1? Group 1 are not given any cells. So there is no immunity for that mice. Naturally it will die. So that makes A correct. Give 1 minute. A correct means okay. In all options A is correct. Okay. Now group 2 mice are likely to produce antibodies in response to polysaccharide antigens. Now what was group 2 given? Group 2 was given thymus derived cells from syngenic donor. Now what is this syngenic donor? That is when you transplant cells between genetically identical individuals. That is just like your identical twins. They have the same genotype. So when you do transplantation between genetically identical individuals, it is syngenic. Okay, now they are given group 2's mice are given thymus derived cells. Thymus derived cells means only T cells are there, B cells are not there. So, and 2 months later immunized with polysaccharide antigen. Now the thing with the polysaccharide antigen is they directly stimulate the B cells to produce antibodies. Now group 2 they are not given B cells, they are given only T cells. So antibodies will not be produced by group 2 mice. So what they have told in B, group 2 mice are likely to produce antibodies in response to polysaccharide antigen is wrong. They will not produce antibodies. That makes option 1 gone, 2 gone. Okay, 3 and 4 we have to see. Then group 3 mice are likely to produce antibodies in response to polysaccharide antigen. What have they done with group 3? Group 3 was given bone marrow cells. Okay, that means both B cells and T cells are produced from a syngeneic donor and 2 months later immunized with a polysaccharide antigen. Okay, B cells are available. Polysaccharide antigen can stimulate the B cells to produce antibodies. So group 3 is going to produce antibodies. So C is correct. Now we have to see if D is also correct. Group 4 are given bone marrow cells. Okay, B cells and T cells are produced from a syngeneic donor and 2 months later immunized with a T-dependent antigen. Now a T-dependent antigen means it requires T cell cooperation with B cells to produce antibodies. Okay, that is T cell cooperation with B cells to produce antibodies. Now group 4 they are given bone marrow cells. So T cells and B cells are produced. The T and B cells can cooperate together to produce antibodies. So what have they told here in D statement, that is group 4 mice are likely to produce antibodies in response to T dependent antigens is correct. So that makes option 3 wrong and 4 correct, that is A, C, D are correct. Now this is with regard to aging. Following statements were made regarding regulation of aging in C. elegans. So in most cells P53 remains, okay, now before that I will tell you the pathway. 1 minute. Okay, so this is the pathway. This is a insulin-like ligand that is binding to its receptor DAF2, and DAF2 is an insulin receptor ortholog. We already learned with regard to FOXO and insulin just 2-3 questions back. Now here also in the C. elegans it is having an insulin-like receptor that is DAF2, and it will be bound by something like insulin but not exactly insulin. And there is a homolog of FOXO here, and full form of FOXO is fork head transcription factor. Okay, so there is a homolog of FOXO here, that is DAF16. Similar to how insulin we saw how insulin regulates the FOXO entry exit from nucleus and all, the same way here also by phosphorylation with AKT. Okay, so that's it. The rest of it I will tell you as we read the questions. Okay, now first one. In most cells, P53 remains bound to a repressor protein to keep it inactive. Okay, P53 is found in humans, and C. elegans ortholog of P53 is CEP1. Okay, so P53 it binds to the repressor MCM2 in humans to keep it inactive, and which is activated under oxidative stress and DNA damage. So oxidative stress and DNA damage, they are linked to aging process. So whenever the cell sends oxidative stress and DNA damage, P53 gets activated. How? By separating from the repressor. Then active P53 it participates in DNA repair, cell cycle arrest, and thereby influence longevity and aging. So A is correct. So that means second gone, third also gone. So 1 and 4 we have to see. DAF2 function as insulin-like growth receptor to block forehead transcription factor and increases the lifespan. Okay, now see here. DAF2, okay, insulin-like growth factor receptor. Now when insulin is bound, AKT is phosphorylated. AKT phosphorylated means DAF16 is phosphorylated and it will prevent entry. Now DAF16, it activates the gene that extend lifespan. It is a transcription factor for such kind of genes, that is that extend lifespan. So if DAF16 is not entering the nucleus means aging is happening and the elegans can die. So it increases lifespan here, as per me, is a wrong statement. It decreases lifespan. But in your answer key, they have shown A and B as the right answer. But as per me, B cannot be the right answer. I have checked research articles also. There also I found that it doesn't increase the lifespan, it decreases. So if you have anything regarding that option, whether I am wrong, you can put it in the comment section. Okay. Now when DAF2 is not active, okay, DAF2 insulin-like receptor, then cells reduce the production of DNA repair enzymes. No. Reduced DAF2 means DAF16 is not phosphorylated. It can enter the cell. It can result in increased activity of the DNA repair enzymes. So DNA repair enzymes are more active means longevity increases. So reduced production, no. So C is not correct. Dietary restriction increases mTORC1 activity, enhancing functional stem cells and longevity. Okay. Now dietary restrictions, they decrease, see here, I have put this inhibitory mark, mTOR activity. Now there is something called as autophagy and it result in promotion of longevity. Okay. Now HLH30, this one is a transcription factor that regulate the gene expression of this genes involved in autophagic process. Okay, expression of genes involved in autophagic process. So in a nutrient rich environment, mTOR gets activated. This one is activated and this results in the phosphorylation of HLH30 and inactivation of it. So HLH30 will not be able to enter into the nucleus and it will not result in expression of the genes involved in autophagic process and increasing longevity. Now when the nutrient levels are low, that is dietary restrictions, mTOR is inhibited, HLH30 can enter the nucleus and activate the transcription of genes essential for autophagy. So here what is wrong is dietary restriction increases mTORC1 activity. No, it inhibits mTORC1 activity. So D is also not correct. As per your exam key, answer key, A and B are correct. As per me, B is not correct. I told you already. So that's it.